Hi, this is Rochelle Crosbie Watson, and we're here today to talk about how utrophin is a compensatory protein in Duchenne muscular dystrophy. So let's start with a case study. We've already learned that utrophin is a dystrophin paralog and is expressed around the extrasynaptic sarcolemma in MDX muscle and in DMD muscle. How would you test the hypothesis that utrophin can compensate for the loss of dystrophin in MDX muscle? Remembering the underlying component of this question is that the MDX mouse, which is a genetic model for Duchenne muscular dystrophy, does not display the severity of symptoms that we've seen in Duchenne muscular dystrophy patients. So, if you suspect that the upregulation of utrophin in dystrophin deficiency protects the MDX mouse from the severity of disease, then the way to address that would be to create an MDX mouse that is lacking utrophin. So we're removing that compensatory component and asking what happens to the disease outcome. And these were the exact experiments that were performed by Kay Davies to really nail down whether utrophin compensates for dystrophin deficiency. So to conduct those experiments, they took MDX mice, which have mutations in the dystrophin gene, and they crossed them with mice that lacked the utrophin gene. So these were genetically engineered mice that lacked utrophin. These mice are fairly normal, the utrophin null mice, except they have some defects in neuromuscular junction structure. So these two mice were crossed, and they generated mice that were MDX mice, so lacking dystrophin, and also mice that were lacking utrophin. So these mice are frequently referred to as the double knockout mice or the DKO mice. And what do you think the phenotype of these mice would be if utrophin compensates for dystrophin? Well, the results of that are shown here. The dystrophin double knockout mice show many signs typical of the human form of disease of Duchenne muscular dystrophy. And dystrophin and utrophin have complementary roles in normal muscle function, is what they concluded from their studies. So you can already predict that these double knockout mice show very severe symptoms of disease. Because we've looked at how to analyze mouse models of muscular dystrophy, I think we have a pretty good idea of some of the outcome measures that this group used to analyze the double knockout mice and compare them to just the single knockout dystrophin deficient mice. And those results are shown here. So we're looking at normal mice, which are about the same size as MDX mice. So the normal mice aren't actually pictured here, but they are the same size roughly as the MDX mice. And compared to that is the double knockout mouse. And I hope you can see that the double knockout mouse is much smaller than the MDX mouse. So already we know that removing utrophin has a detrimental effect on the growth of the MDX mice. You can also see the kyphosis, or the curvature of the spine, that results in the double knockout mice. This results from skeletal muscle weakness. Looking histologically at transverse skeletal muscle cryosections, you can see that there are some signs of pathology in the MDX mice, but it's really exacerbated in the double knockout mice. You can see evidence of muscle damage, of necrosis. There is significant fibrosis. So the pathological state of the double knockout in an age-matched manner is much, much worse. There is also a hind limb clasping defect. So wild-type mice, when picked up by their tail, tend to splay the hind limbs like this. But the double knockout mice are actually very crossed, or the hind limbs are very close together. This is an abnormal hind limb clasping event, which is very evident in the double knockout mice. So these mice are quite severe, and in fact, their age is severely shortened. The double knockout mice die anywhere from 9 to 13 weeks of age, whereas MDX mice live a relatively normal lifespan, very comparable to that of a wild-type mouse, which is around 2 years of age in a laboratory setting. So this diagram is showing you a really great comparison of the double knockout mice, and also an experiment that we're about to get into in which utrophin is overexpressed in the double knockout mice to show that it can compensate both for dystrophin and utrophin deficiency. But this is a really great image in showing you the effect of knocking out both dystrophin and utrophin in a mouse model. So Kay Davies established, using these double knockout animals, that utrophin really seems to compensate for the dystrophic phenotype in the MDX mouse model. That's very suggestive of a functional redundancy between dystrophin and utrophin. They wanted to carry these experiments one step further. If utrophin could compensate for dystrophin, if they have very similar protein structures, they both bind actin, although slightly different manner, could we bump up and really drive utrophin expression in the MDX mouse to even more greatly increase the interaction with laminin and to more greatly protect the sarcolemma from contraction-induced injury? So they did those experiments by creating first a utrophin transgenic mice. These mice are pumping out lots of utrophin protein in skeletal muscle, and they cross those with the dystrophin deficient model, the MDX mouse, to produce a mouse that lacks dystrophin but is expressing lots and lots of utrophin. And they wanted to ask, could this be a potential compensatory effect? Could the increase in utrophin protect against disease? So the results of that were published in a really great Nature Medicine paper, and they found that sarcolemma localization of a truncated utrophin transgene in the MDX mouse significantly improves the dystrophic mouse phenotype. They demonstrate for the first time that expression of utrophin in the MDX mice really prevents the symptoms and the development of muscular dystrophy. So here's a case study. You've just learned how Davies and colleagues created a utrophin transgenic MDX mouse. Where do you think the transgenic utrophin would need to be localized within the skeletal muscle cell to prevent muscular dystrophy? And can you predict how this would affect the skeletal muscle phenotype? Let's take a look. So the answer is shown here. Let's take a look at the data. Okay, so this is an immunoblot analysis. And what we're looking at here is the expression and the abundance of utrophin in various tissues taken from 3 lines of utrophin transgenic mice. So when Davies et al. Did these experiments, they created 3 separate lines of transgenic mice: the fur, the free, and the Fiona. These mice differed in the levels of utrophin overexpression. For most of their studies, the group focused on the Fiona transgenic line because it seemed to give the greatest rescue of the skeletal muscle. But what you can see here is that there's a great increase and a great expression of utrophin in these utrophin transgenic MDX mice. Now, if we look at where the utrophin is localized in MDX mice that are overexpressing utrophin, I think you can already predict that if this protein is preventing the disease from happening, then it must be located around the extrasynaptic sarcolemma where it can protect the membrane from contraction-induced injury, and that is indeed the case. So what we're looking at here are transverse skeletal muscle cryosections that have been stained with antibodies that recognize utrophin in the upper panels and a utrophin binding protein called dystrobrevin in the bottom panels. So you can already see in the MDX section that there is some faint staining of utrophin around the sarcolemma, and that's dramatically increased in the 3 different transgenic lines that the Davies et al. Group created. And the Fiona line has a really robust expression of utrophin around the sarcolemma. Not only is there abundant expression of utrophin, but that a utrophin binding protein, dystrobrevin, is also restored to the sarcolemma upon utrophin transgenic overexpression. So we have just established that utrophin is abundantly overexpressed in the utrophin transgenic MDX mice. But what are the histological and physiological outcomes of that overexpression? We're going to look at that on the next couple of slides. What you're seeing here is the central nucleation. Remember that central nucleation is a marker for degeneration and regeneration, and that those regenerated fibers have nuclei that are at the center of the myofiber. So if we look at wild-type animals, we'll see that they have very, very, very low levels of central nucleation. There isn't much regeneration that's really needed in wild-type muscle. The MDX mice, on the other hand, which are in yellow, have very high, 60% of the myofibers are undergoing active regeneration. If we now look at the other three utrophin transgenic lines, you see that the reduction, there's a very significant reduction in the overall central nucleation when that utrophin transgene is overexpressed in an MDX background. And in fact, the greatest reduction is the Fiona transgenic line, the FIO, which really almost brings the central nucleation back to wild type levels. So that's quite impressive. So next, we're looking at the muscle mechanics, specifically of the diaphragm muscle. On the y-axis is plotted normalized force, and you can see that the wild-type animals produce robust normalized force from the diaphragm muscle, and that the Fiona transgenic mice are very similar to wild-type mice. Take a look at the MDX mice. Notice that they have very low normalized force levels in the diaphragm muscle, and that the other two transgenic lines are somewhere in between. Also, an important feature of dystrophin deficiency is the susceptibility to contraction, eccentric contractions, and also the susceptibility to sarcolemma membrane damage. And you can see in these experiments that the MDX mice are very susceptible to membrane damage and show a dramatic drop in force in eccentric contractions, and that those are all restored to near or at wild type levels in each of the three utrophin transgenic lines. Lastly, if we look at physical activity of these different genetic mouse models after a mild treadmill exercise, you can see that the physical activity of wild type mice is very high after a treadmill exercise event. They're not deterred by the exercise activity, and that is very much suppressed in MDX mice, but again, those activities are restored for the utrophin transgenic mice. So, what I hope that you've learned from this second mini lecture is that utrophin is structurally similar to dystrophin. They both interact with actin, and that utrophin can compensate for the loss of dystrophin activity. This is the gorgeous, beautiful basic science groundwork that went into supporting utrophin as a therapy for Duchenne muscular dystrophy. It really addresses the primary loss in Duchenne muscular dystrophy muscle, which is the susceptibility of the sarcolemma to membrane damage. There are many, many other compensatory proteins in Duchenne muscular dystrophy, but utrophin is the one that is really the most well characterized, which is why we've presented it here today.